Okay, this is the next lecture on mitosis. So this is the M phase of the cell cycle. So after the DNA has copied itself, after it's been replicated during the S phase, it's then going to be separated. The two copies are going to be separated during mitosis. And so that's what we're going to focus on in this lecture, the M phase of the actual cell cycle here. So in the G1, you have one copy of the genetic material. During the S phase, two copies have been made. And now we're going to focus on how, in this M phase, those two copies get separated into two separate cells. So the first thing that's going to happen is this DNA molecule is going to form a complex with some proteins. It's going to wrap around and associate itself with proteins to form what we call chromatin. Chroma means color, so you could actually, when they looked at a nucleus of a cell, they could see a darkened area, and that's where the name chromatin came from. So this is how the DNA kind of gets packed up during this process. So the DNA itself is very, very long, and if you had all your DNA stretched out, it wouldn't fit into a cell. So what we find is that it's in this compact, condensed form. And it's during this, like this, even during interphase, it's just to a much lesser degree. And this is actually, we'll learn later, a way of controlling whether genes can be expressed or not, because when it's tightly coiled, genes are not able to be expressed. So what happens is we have our long strand of DNA, and it starts to coil upon itself, and then that will wrap around these proteins called histones. And then we get more of them wrapping around and wrapping around, wrapping around, wrapping around, and we get these little like bead units called nucleosomes. And so now these nucleosomes, they start to coil and tighten up and wrap up, and we end up with what we call chromatin. So the chromatin is going to be much thicker than the original DNA, and it's going to be much shorter than the original DNA. And that's what you're able to see under the microscope. Okay, so now we have our coiled up DNA, these little structures called chromosomes, and now we need to separate them. So what's going to actually separate them are these, this what we call a spindle. So this starts with these centrosomes. So a centrosome is a region of the cell that contains these centrioles. And centrioles are where these spindles, or these microtubules, are going to start to form their complexes. Centrioles and centrosomes are not found in all cells, but a centriole, it would kind of be like if I took a handful of pencils in one hand and took a handful of pencils in the other hand, and I put my two hands at right angles. It's kind of how the centrioles are. So from the centrioles, these microtubules form. And these microtubules come out from the centrioles, and they attach to the chromosome at the centromere, or at the place where they're attached, connected to each other during the beginning of cell division. And this side has another microtubule that's going to come out and attach to it. And then there are also microtubules that are going to come out and attach to other microtubules from the other side. And these are going to ultimately change the shape of the cell. The cell actually elongates during this process of cell division. So we have the kind of the components. We have our chromosomes and how they're formed. We have our spindle and how that's formed. And so now we're going to look at how the chromosomes actually get separated. So here's a cell in the beginning when it's just during interphase, so there's none of this mitosis is occurring. We have the centrioles outside of the centrosomes outside of the nucleus. Here's the nuclear membrane. Inside of the nucleus is the nucleolus. And so in the beginning of mitosis, what's going to happen is these chromosomes are going to start to condense and condense and condense and condense to the point where you can actually see them. And you saw this under the microscope. It's kind of how you identified that prophase was beginning, was that you were able to start seeing these like squiggly things inside of the nucleus. Now the centrosomes are moving from pole to pole and separating, and this is ultimately going to determine the direction of this whole process. And now the chromosomes have coiled up into the point where we can actually see individual chromosomes. And each of these are duplicated chromosomes. Here's one copy, and here's another copy of the exact material that happened during the S phase. And then that nuclear envelope, nuclear membrane, is going to break down, and our spindle is going to be forming and attaching to our chromosomes. The next thing that's going to happen is, remember, you have a spindle fiber microtubule coming out on the side and attaching to the chromosome at the centromere. We have one from the opposite side coming in and attaching to the centromere. Well, they kind of play like a tug of war. And these chromosomes get pulled back and forth, back and forth, back and forth, until they're all lined up on this invisible line in the middle of the cell. And there's actually a checkpoint for that as well. So that it won't go to the next phase unless all of the chromosomes are lined up properly. And then suddenly, the centromeres, the protein, the kinetochore that's attaching them at that point, breaks down, and the sister chromatids separate from each other and get pulled to opposite poles of the cell. And then everything that happened during the beginning is going to kind of go in the opposite direction. So the nuclear envelope is going to reform. The chromosomes are not showing you this, but they're going to uncoil. And our cell and our nucleolus is going to become visible again. And then our cell, if it's an animal cell, is going to pinch in, kind of like purse strings. Microfilaments are going to pull it apart until you end up with two separate cells. You do not need to know the names of the phases of mitosis, but you do need to know the movement of the chromosomes. Okay, so what's holding these chromosomes together is this cohesin. So this cohesin is formed when it's the DNA is copied. So remember, this is one copy, this is another copy. We call them sister chromatids. And during G2, this is how the DNA is structured. And then in the beginning of mitosis, or in metaphase actually, the cohesin breaks down, and you're left with just the cohesin at the centromere. And then when the chromosomes are going to separate, it breaks down at that point as well, and then you're left with two separate chromosomes. One little tricky thing, and I don't know if they're going to ask you a question like this. They used to like to ask these questions. This is one chromosome. I would call this a duplicated chromosome. Over here, these are two chromosomes. They're no longer attached at the centromere, and they would be considered their own little chromosomes. Over here, because they're still attached, we would refer to these as one chromosome, even though there's two copies of it. So I like to call this a duplicated chromosome, and this just a chromosome. Alright, plant cells, because they have a cell wall, it's a little trickier, so they can't just pinch in like animal cells do. They have to form that cell membrane, but they also have to form a cell wall. So what they do is vesicles coming from the Golgi apparatus are going to kind of come together at the center of the cell, and they eventually fuse, and these are going to then form this, what we call a cell plate. And this cell plate is ultimately going to turn into the cell wall between the two cells. Okay, I want to talk a little bit about apoptosis. We've talked about this before, and it's on the new curriculum several times, so I thought it would be worth kind of just touching on it again. So we're talking about how we make new cells, but we also have needs to break down cells or get rid of cells that are no longer needed. And so this process is done by apoptosis. And apoptosis is kind of like cell suicide, and different things that can cause a cell to go through apoptosis is during embryological development, there are parts that are formed that are then no longer going to be needed. For example, I don't know if you know, but your fingers were webbed at one point in your embryological development. By the time you're born, that's not the case anymore, and that's because the connective tissue between the fingers went through apoptosis and was broken down. If a cell becomes cancerous, if it gets enough genetic mutations that it could become cancerous, it may end up going through apoptosis. Cells that become infected with virus might go through apoptosis. Your blood cells do not even have your red blood cells don't even have a nucleus. They only live about 120 days. Your epithelial cells or your skin cells are included in this. They don't live for very long, and they'll die after a few days. So how these cells are destroyed is through this process of apoptosis. And this is just a little picture of that. So first of all, cells tend to like to be together, and one of the things that will happen here is that the cell that's going to go through apoptosis will separate from its neighbors. And then once it separates, it will then chop up its chromatin. So those little nucleosomes, those little DNA wrapped around those histones and those little bundles, they'll get chopped up into those separate little bundles. And then it forms, they call them blebs, little outcroppings of the cell, which then fragments. And then finally, cells in the vicinity that are involved with phagocytosis will come along and destroy the parts that are remaining. And your body is very good at recycling materials, so that stuff will be used. Okay, you're doing a project on cancer, but I want to talk just about a couple of things with cancer. So here we have cells that are normal, and if we put cells on growth medium, and remember cells need some kind of a signal in order to divide, so we'd have to put something in like a growth hormone, a growth factor onto the plate here. So there's some nutrients and some chemical that's going to signal cell division. The cells will divide, and they will form a single layer where the cells are all attached to each other along the bottom of the dish. And then if we came along and we removed some of those cells, the cells would then replace those cells. So the division in that area would fill in that little hole, and then they'd stop again. That's normal. If we try to grow cells that are not normal in a Petri-like dish like this with growth factor, what we end up having happen is the cells will grow right on top of each other. They don't follow these rules. And the other thing that can happen is here the cells are all attached to each other. These cells can actually break free and go move to somewhere else and start dividing, and that's called metastasis. Okay, so there are two proteins or two genes that code for proteins that have been identified in all the cancer research. So they're kind of misnamed in a sense because they were named by oncologists, people who are studying cancer. So the first is an oncogene. And an oncogene is opposite of, we talked about the tumor suppressor gene. And oncogenes are kind of, they turn on cell division. So if a oncogene is normal, it's going to turn on cell division when cell division needs to happen. But if something happens to that oncogene where it becomes overactive, you can end up with this cell division being turned on when it shouldn't be, and then you can get cancer. The other genes that are involved in this are tumor suppressor genes, and these have the opposite effect. So we talked a little bit about P21. The retinoblastoma gene or protein is the one that was in that restriction site. That was the protein that was phosphorylated, and that turned it off. That was also a tumor suppressor gene. There's another one that's very common called P53. You may find these in your research when you're doing your cancer project. But these are ones that are going to turn off cell division. So when these are not working properly, then cell division can happen when it's not supposed to. So these will block cell division until cell division should occur. But if we block these for some reason, such as the virus that causes cervical cancer, that's the human papillomavirus, that actually blocks the tumor suppressor gene. So again, these are kind of misnamed because they're named by, with cancer research. They're normal genes, but when they're not functioning properly, you end up with cancer, or can end up with cancer. Okay, I have one question with this one for practice, and that is, a human stem cell contains 46 chromosomes in its nucleus. How many chromosomes go to a single spindle pole during mitosis? Okay, and the answer here would be 46, because once we go through the point where the chromosomes separate and the sisters are no longer together, they are full-fledged chromosomes. So even though at the beginning of mitosis we start with 46 chromosomes, we start with 46 duplicated chromosomes. And in the end, we end up with 46 in each side of the 46 chromosomes on each side of the cell, and then that's going to split in half. And you'll end up with two cells with the 46 chromosomes that you started with. They just will not be duplicated going into the G1 until they get into that S phase, and then they'll be duplicated. So that's the last slide for this. I'm planning on doing one on DNA replication. I may wait to see if you guys are using this or not, but thanks for watching, and take care.